Okay, so I think we can start. Hi everybody, and thank you for joining again for the Museum Restart, a series of meetings dealing with changes and opportunity following global pandemic. The series were created in partnership between ICOM Israel, the US Embassy, and the Smithsonian Institution. I'm Efrat Haberman, and I'm the coordinator of the Collection Manager and Registrar's Forum of ICOM Israel. And today is the last session we are going to deal or focus on the collection. Well, the pandemic affected collection management in all sorts of ways. And at our previous sessions, we discussed about the way the collecting activity and methods during historic events and especially during COVID-19. And today we are going to see it from another angle and to hear about how a scientific museum collection could use for research or studies. Pamela Soltiis will talk about the way museum collection could help fight the next pandemic. And Pamela is an American botanist. She's a distinguished professor at the University of Florida. She's a curator at the Florida Museum of Natural History, principal investigator at the Laboratory of Molecular Systematic and Evolutionary Genetic at the Florida Museum of Natural History, and founding the director at the University of Florida Biodiversity Institution. During a lecture, you can leave a question at the chat, and we will have time for question and discussion at the end. So, Pamela, thank you very much again for joining us today, and the floor is yours.
Thank you. Well, thanks so much, Efrat, for the introduction, and thank you to yourself and to ICOM for putting this seminar series together and for including me. I'm very honored to participate. I'd like to focus today on how natural history collections can help us address major societal problems. And I will give the example of the pandemic, but a number of other examples as well. Now, I'm coming from the perspective of natural history museums, but I think all museums share some fundamental principles related to preservation and interpretation of their specimens, artifacts, and objects. So despite the fact that my title is oriented toward pandemics, my research expertise and interests are really in plant evolution. So many of the examples that I'll share today relate to plants, but I eventually will get to the topic of the pandemic. Now, many of the themes that I will be addressing today are also presented in this consensus report on biological collections, which was published last year by the US National Academies of Science, Engineering, and Medicine. And I was fortunate to have served on the committee that produced the report, and I'd like to acknowledge the amazing committee members I worked with, because some of the ideas that I'm presenting today undoubtedly emerged through those discussions. Now, natural history collections our specimens have been collected for centuries. And these pressed plants, as shown here, represent examples of these natural history collections. Plant specimens such as these are housed in special collections called herbaria. These are essentially plant museums, and they occur worldwide. In fact, every one of the world's natural history specimens has a story to tell. And what's more, each specimen is packed with data that can be used in a broad range of scientific studies. So today I'd like to provide an overview of natural history collections, an introduction to the status of digitization of these collections, and then provide some examples of how we can use natural history collections to address societal challenges. Now, there are between 1 and 2 billion natural history specimens in the US and somewhere between 3 and 4 billion estimated worldwide. In the US, these are housed in about 1,600 natural history collections. Worldwide, we're not quite sure how many there are, although there's a developing registry that is helping to connect all of these collections. But there are certainly somewhere over 3,000, maybe over 4,000, or maybe even more natural history collections worldwide. Now, most specimens were originally collected as a way of documenting the biota of a particular region. And for most of their history, museum specimens have formed the basis of the fields of systematics and taxonomy, as they still do today. So these are the fields that are focused on identifying and classifying and understanding basic patterns of biodiversity. And most of what we know about the living world actually comes from our natural history collections. So this shows Carl Linnaeus, or Carolus Linnaeus, the father of taxonomy, as a young man with all of his collecting gear and a specimen named in his honor, Linnea borealis. And again, collections, these in London at the Linnaean collections of the Linnaean Society. So although fundamentally these specimens have been used in these fields, in fact, today the uses of these collections extend far beyond these traditional fields. So today, natural history collections are used in the study of genetics, genomics, chemistry, species interactions, phenology, biogeography, and even more. And these amazing photographs from the collections of the Smithsonian really illustrate the extent of some of these very large collections, showing butterflies on the left and various shelled organisms, mostly mollusks, on the right. Now, one of the issues with our natural history collections, though, over the years is that most specimens have been locked away in cabinets and are unavailable for general use. And so it's really only a specialist who is able to travel to one of these large collections, or perhaps request a loan to one's own institution, that have been able to use these collections. And that means that the actual application of these amazing collections have been quite limited, at least with regard to their potential. But digitization is changing that and is bringing specimens out of the cabinets and onto computer screens worldwide. So what do we mean by digitization with regard to natural history collections? I know that museums of all types are undergoing various types of digitization, with databases being developed and various sorts of images being developed. In natural history collections, we use digitization to refer to both data and images. So each specimen, although in this case a pressed plant or part of a plant, actually carries important additional information in its label. And this is true whether it's a plant specimen, a fungus, a vertebrate, or whatever. Now, ideally, the label, which for a plant specimen is typically located in the lower right inside that circle, will contain the scientific name of the species, the date that the specimen was collected, the name of the collector, the location of the collection. And in the US, this would include the state, the county, a specific description, and with more recent collections, also GPS coordinates. In addition, any associated species that occurred with the specimen would be noted, and there might be other notes about the habitat and so on. And basically, the more information that's provided either on this physical label or in a notebook, the better. So as you can imagine, a detailed record can provide really abundant data. And today, this information is being databased so that there are electronic records that are associated with our specimens. And in addition, images of various types are providing access to the specimens while also revealing new aspects of the specimens themselves. So for example, in this slide, I showed this just regular photographic image of the plant specimen on the right, but there are other types of images that are shown for other organisms. So applications of various types of X-ray applications, also different sorts of imaging that allow for coloring in of different structures and different tissues and different sorts of materials, or and most recently, as on the bottom, CT scanning. And this can provide information, for example, in a vertebrate, this is a frog, information about the skeleton, and also in some cases, information about soft tissues as well. So a whole range of imaging technologies is being applied to natural history collections as well. And when we put these images together with the information, that's the label information, we start to build a very rich record of information about each specimen. Now, in the United States, the national center that coordinates the digitization or databasing process and then compiles the data from all of our natural history collections in the country and provides this information to the public is called iDigBio. And it currently contains over 131 million records of natural history specimens, about half of which are plant specimens. It also contains over 42 million media records, and these include images, vocalizations, videos, and so on. But of these 42 million, about 36 million are images of plant specimens. I think you can imagine that being flat, like these pressed plants are, and being glued onto a regular sized piece of paper makes these herbarium specimens particularly amenable to photography imaging and then inclusion of those images in databases. Other sorts of organisms, such as frogs that are in jars of liquid preservative, are much more difficult to image because each one has to be taken out of its jar, positioned, photographed, etc. So most of the images available worldwide today are of plant specimens, which for a botanist is a great thing to see. So if you go to the iDigBio website, which is idigbio.org, you'll see a map that shows the density of specimen coverage. And as you can see, specimens cover essentially the globe, both terrestrial regions as well as marine regions. Now, for reference, in some of my talk today, I'll focus on some of the aspects of the biota of Florida, the US state of Florida, which is marked by this turquoise circle on the map here, or when enlarged here. So just to provide a little bit of geographic reference. Now let's return to the specimens and our occurrence records. So as we've seen, each specimen can carry valuable information. So for example, we can search the iDigBio database for any given species. And let's say we'll search for the plant called the red maple tree. And we can enter that into the search criteria on the left part of this image here. And if we do a search, this will return to us a map with a number of points, with each point referring to a single specimen and providing a snapshot of the information that's available for that specimen. In addition, some specimens don't have the geographic information that allow them to be plotted on the map. So there would be additional records available, but just not visible on the map. So we can click on one of these dots and we can pull up part of the record, and then we can pull up the full record, and this might indicate that there is actually an image associated with that locality. So for each specimen record, the record represents a single location on a map. Again, the green dots in the center map. And if we use other databases, we can actually gain information about the ecology of that site. So that specific location of each of those dots can be investigated or queried through other databases. And we can learn something about perhaps the temperature at that site, the precipitation, the soil type, the soil chemistry, elevation, other specimens that have been collected from that same location, and so on. And so that physical location on a map actually is an entry point to much extensive information on the ecology of each specimen. So this just illustrates some of the capabilities of iDigBio, again, the national center in the US, and it's actually centered at the University of Florida, and I'm one of the directors of this project. But there are similar sorts of aggregators in other parts of the world. So there's a national specimen information infrastructure in China. There's a structure called Canadensis in Canada, the Atlas of Living Australia in Australia, Species Link from Brazil, and a number of other smaller such infrastructures. Some of them are also taxon specific. So there's another one called VertNet, which focuses just on vertebrates. But in all, these separate aggregators provide a particular orientation for the data that they have databased. And all of them lead into the Global Biodiversity Information Facility, which is a global data aggregator that is funded by the countries of the world to provide biodiversity information. Now, one of the most fascinating aspects of natural history collections is that these specimens have unanticipated uses. The specimens were collected for one purpose, probably to document the biota of a given area, but over time, they've been used for many other sorts of questions that the original collectors, in most cases, could not have even envisioned. And this point argues strongly for the value of our collections and for long-term investment in their care. So for example, natural history collections are now being used to address a number of biodiversity-related societal challenges, such as the effects of global change on biodiversity, including the loss of biodiversity itself, the presence and patterns of invasive species, changes in land use, loss of ecosystem services. And by ecosystem services, I'm referring to such things as the maintenance of clean water, soil stabilization, flood resistance, pollination for crops, sources of new foods and medicines, beauty and relaxation, and so much more. And together, these services that natural ecosystems provide are worth hundreds of billions of dollars annually. But because of various sorts of human-mediated changes to our environment, these ecosystem services are being lost. In addition, our collections can help us prioritize and establish good strategies for conservation. They can be used in addressing issues of food security. And as the title of my talk actually focuses, we can learn about emerging pathogens and other factors related to human health. So today I'd like to share a few examples of how natural history collections are being used to address societal problems. So specimens, even if they were collected a century or more ago, can be used as resources for computational modeling. They can be used to predict the impact of climate change. They can aid in conservation. They can help us understand, mitigate, and prevent pandemics. And they can contribute to the design of materials and robotics. And today I'll briefly explore each of these applications. So let's start with modeling the responses of species to climate change. And this is a type of work that's being performed for many groups of organisms. And the examples that I'll show here are for plants and plants from Florida. Now, using the ecological information that's contained in or that can be inferred from the specimen record, we can develop models to characterize the ecological niche or conditions for a species. And we can use these models to evaluate the potential response of a species to climate change. And I'll share just a couple of examples. The first of these is called the flat spike sedge, or the scientific name is Albigardia ovata. And this plant is a relative of the grass family. Its ideal habitat in the state of Florida is shown by these warmer colors. And you can see them here sort of in the Miami region of Florida. Now, this ideal habitat has been modeled and from the ecological information that's present and associated with with the locality of the specimens. But where will those ideal conditions be in the future? So we can take the conditions that we know are ideal for the habitat for this species today, and we can ask, where will those conditions of temperature, precipitation, and soil characteristics be in the future? And when we do that experiment, we see that, in fact, there is very limited, if any, suitable habitat for this species just within the next 30 years. So in this projection of the models, we don't see any yellow or red colors which signify the most suitable habitat. Instead, there's just this very light green in the south part of Florida, suggesting very limited, if any, suitable habitat. So this suggests that within the span of just 30 years, the conditions will change to such an extent that this species will have a very hard time finding any suitable habitat. And this is a species that only occurs in southern Florida today. It's restricted to that habitat. So in 30 years, without some changes, this model suggests that the species will be extinct. Well, how common is that sort of expectation, or are other outcomes likely? Well, we can look at another species. This one is called the scrub plum, and it occurs natively in what we call the high elevation area of central Florida. It's a ridge that runs sort of through the backbone of the peninsula. And in fact, when we do our modeling, that is exactly where the models indicate that it should occur, right here in the central part of the state. So when we project the models onto the expected conditions for 2050, we actually see something quite surprising. We see that there's a lot of yellow, orange, and red in this map, suggesting that there is likely to be a lot of suitable habitat for this species in the future. And that's because, of course, the temperature is increasing, and the precipitation will, in most cases, be decreasing. So this is a plant that likes it hot and dry, and that's what the conditions will be like in the future. Now, of course, it's hard to imagine a species that currently is restricted to a very small area in the central part of the state to actually be able to disperse and cover all of these potentially very viable habitats. But it's certainly possible that there will be good habitat available for this species in 2050. So for some species, the outlook is quite dismal, for others, quite positive. So we applied the same modeling approach to about 1,500 species of Florida plants. This is about 1/3 of all of the plant species that occur in Florida, which is a very highly diverse, biodiverse region. And we can start out with a map that looks like this. Here, the green regions show high species diversity today based on these models, and the tannish brown regions show low species diversity. And then we can ask what will happen in the future. And we can look at a map for 2050 and plot the number of species in 2050 minus the number of species now. In other words, we can look at the changes in these regions. And we'll see the areas that currently are very species rich, like this dark green area in sort of north central Florida, actually will be losing species. And the areas such as the northern border of Florida, which has more green to it in the future, will be gaining species. So this is the type of response that we would expect for temperate species, that as the climate gets warmer, species will move northward. And that's what is predicted for most of the plant species in Florida. However, we see actually some other interesting situations where other species appear to be having more suitable habitat to the south in this region here in the southern tip of Florida. So if plants could actually move to the extent that the models suggest where their suitable habitat is, we would see a northern movement and a southern movement. Now, the slightly concerning aspect of this, one of many anyway, is that this area in South Florida is the area that's mostly covered today by the Everglades, a very large swampy region. And it's unlikely that that will actually have the right soil characteristics and water characteristics for these terrestrial plant species. In addition, it's a very low elevation region of Florida, and many of our models of sea level rise suggest that this part of Florida will actually be underwater certainly by the end of the century, if situations are not changed. So what this suggests is that the plant diversity and its distribution in Florida over the course of the next century will be very likely to change relative to today. And this just shows a problem that's a global situation, but it shows it in a microcosm on a much more local scale. Now, these models can also provide us with some information about how to establish conservation areas. There's a lot of discussion about conservation. What do we protect? Do we protect the rarest of rare species? Do we protect regions that contain a lot of species? Do we protect so-called biodiversity hotspots? These are the regions, for example, in California here, a few here in Northwest Florida, and these regions in the Appalachian Mountains that show, that are marked with red, that are these very high species rich areas. But what do we protect? Well, there are a number of different ways that we can use these models to evaluate conservation priorities. So, for example, this map shows relative amounts of what we call phylogenetic diversity. We can just think of it as another way of looking at plant diversity, where dark areas represent high levels of diversity and lighter areas less diversity. And we can ask how well our conservation areas that we currently have capture this plant diversity. And then we can use that as a way of moving forward to ask questions about what other areas might we want to protect. So here I've marked with red areas that are currently under conservation. So all of these green blocks, both on land and in the ocean, represent preservation areas. And here we have some areas that are preserved, but they actually have low diversity relative to this map. And then there are other areas, such as these regions that I marked in turquoise, that have high diversity, but they're not currently preserved. So this suggests that an effective way of preserving a large number of species would be to establish conservation areas in some of these areas with high diversity. Now, just remember that all of this information about these possible prioritizations for conservation actually have come from modeling using information in herbarium specimens, our natural history collections that in some cases were collected 100 or more years ago. Now, climate change is not only affecting the distributions of species, but it's also affecting their annual cycles. And this is something we refer to as phenology. So for plants, phenology refers to such key seasonal changes as bud burst, when the leaves first come out, also when the flowers first appear, and then when fruits appear. And these are patterns that are very characteristic across both North and South temperate regions. There are also phenologies associated with other sorts of climatic regions, such as the tropical regions and both wet and dry tropical areas. And in those cases, the phenologies are perhaps less well understood and less predictive. But in the temperate regions, we expect to be able to see on a predictive basis bud burst, flowering, and fruiting times. But of course, what we're seeing with climate change is a shift in those timings. So this is a map from the US National Phenology Network. And this network tracks patterns and timing of phenological change each year across the country. And this map shows the extent of first flowering for a select set of species. In March of this year, this is what this map looked like. So the darker colors in the south indicate that the first flower occurred longer ago, and the regions in gray here in the north are areas where flowering hadn't yet begun. So we can look at a pattern like this and wonder how this compares with other years. Is this a typical year, or is it an early year or a late year? So we can actually use data in this network to show differences from average. Here, the red areas show earlier than normal first flowering, for example, in the Southwest and up the West Coast, and in some regions of the Midwest. And then these areas in blue, including, well, Texas, parts of the South, up into Virginia and part of Florida, are later than normal. So these maps are compilations of data for the last 40 years, and they look at a specific set of species. And they tell us how plants are responding in terms of flowering time to a change in climate. But what if we could look at more species, and what if we could go back earlier in time, so that we're not limited just to these observations of a set of species over only 40 years? Well, in fact, herbarium specimens allow us to do just that, because each specimen has its date of collection, and we can tell from a herbarium specimen if it was in flower, like this magnolia in the left, or this pitcher plant on the right, or if it was in fruit, like this maple in the center. So herbarium specimens provide data for many more species than what have been mapped out through the phenology network, and over longer periods of time than those observations that have been collected. Also recall that iDigBio has about 36 million images of herbarium specimens. Of course, they're not all from the same geographic region, but that's a lot of data for studying phenological change. But I think you can also imagine that it's a pretty tedious job having to score each specimen by hand. So looking either at the specimen or an image of a specimen and going through and saying, okay, here's the date, here it's in flower or it's in fruit. But fortunately, we don't really have to do this by hand, because methods based on artificial intelligence are emerging. So what if we could apply the tools of artificial intelligence, the same general processes that are responsible for autocorrect on our phones or for self-driving cars? What if we could use these to help us score herbarium specimens as being in flower or fruit? Well, in fact, we can do that. And we've been working with a couple of groups of computer scientists to develop methods for delimiting reproductive structures, flowers or fruits, on herbarium specimen images, so that the specimens can be scored for these phenological traits. Then we can compare the dates of flowering today against those from 20 years ago, 50 years ago, 100 years ago, and so on, and see the extent of change associated with warming temperatures. Now, in general, but not always, plants are flowering sooner and flowering longer, and we're just starting to be able to ask what this really means for an ecosystem. So these AI methods are able to identify flowers, for example, on this California poppy on the left. Each of these colored blocks represents an individual mask that is identified as being a flower. And in the center, this is a branch of a maple. Each one of these colored masks refers to a fruit. And then we have an orchid here on the right, which has many, many, many very, very tiny flowers. And for some species such as this, it will probably be impossible to apply these methods. But for many other species, there is great promise in applying these methods of artificial intelligence. And so through a combination of modeling and using artificial intelligence for phenological analysis, it's possible to be able to get a much better idea about the impact of climate change on any given species. And of course, again, I've mentioned these with regard to plants because these are the projects that I'm working on myself, but similar sorts of studies are being undertaken in other groups of organisms where modeling is being done just like I described, and where artificial intelligence is being applied to look, for example, at insect emergence, butterfly movements, and so on, so that we can have a more holistic view of organismal interactions, again, based on our herbariums and other museum collections. So I'd like to return again to this theme of unanticipated uses of natural history collections. And again, these are specimens that are collected for one purpose and used for many others, including human health. And it's at this point that I would like to take up the example of how specimens have been used and could be used in the study of disease, particularly COVID-19. So in April of 2020, a COVID-19 task force was organized by two organizations in Europe, the Consortium of European Taxonomic Facilities, or CTAF, and the Distributed System of Scientific Collections, or DISCO. And this task force was advertised to the collections community worldwide, and a number of organizations and individuals joined in. So I participated in this task force as a representative of iDig Bio, and it was a fascinating experience. And I'd like to acknowledge the vision of Anna Cassino, Dimitris Korias, and Wouter Adink, who formed this task force, and for the and to all of the task force members for their inspiration, creativity, and dedication. We met at least once a week for a series of several months, and then multiple subgroups continue to meet to this point in time, addressing different ways that collections can be used to help fight pandemics. So our main questions when we convened this task force were, how can natural history collections actually contribute? And at this point, April of 2020, just a month or so, maybe a little more than that, when many areas of the world were really starting to feel the impact, we felt a real desire to try to use our expertise to bring about some good. And so the questions that we thought might be able to be asked were, what is the natural host of the virus, SARS-CoV-2? Where did this virus originate? What's the pattern of transmission? And we recognized that there were important resources that could be brought to bear on these questions. For example, natural history collections that provide the baseline information on potential hosts. There are also genetic resources where we have vast information on viruses, and other genetic resources that could provide information on viruses if they were analyzed. And then there's an extensive literature on the behaviors of bats and other potential hosts, and with information on where these hosts live, how they move, how they congregate with other species, all sorts of information that could potentially lead us to understand something more about the original transmission from a natural host to humans. Now, to understand how all of these pieces of information might fit together, we used something called the extended specimen as a framework. And this is a concept that was described a few years ago in a book edited by Michael Webster, and I'll have a picture of that book a little bit later in my talk. But it's also something that we considered in this National Academy's report as a fundamental aspect for understanding and using natural history collections. So what do we mean by the extended specimen? This really refers to the physical specimen, such as the frog, as you see here, or perhaps a plant specimen, but it's the specimen and all of its derivatives. So what might be those derivatives? Well, the derivatives could be a digital specimen record, so a database record. It could be video or audio. It could be a genotype, so gene sequences. It might be parasites that are actually isolated from the specimen. It could be any of a number of other sorts of extensions here. And then it could also be information such as models, like what I described before. It might be an evolutionary tree showing the placement of that species. And it might be further description of the species in the literature. So essentially, all of these pieces that have used the specimen or parts of the specimen are related back to that specimen. And so the specimen no longer is one single frog in a jar, but it's a whole network of information that reaches out across databases and through time. Now, in order for the specimen concept to work, all of these elements need to be connected informatically. And so we need effective databasing to tie these different derivative parts back to the specimen. And in order to do that, we need robust cyber infrastructure. So what do I mean by cyber infrastructure? Well, cyber infrastructure encompasses the computing systems, the repositories, the advanced instruments, the software, the high performance networks, and also the people that enable or support data acquisition, storage, management, integration, mining, analysis, visualization, and distribution. In other words, it's all of these pieces in this diagram, and it allows us to go from observation on the one hand through this network, this connected network of resources and data to yield a discovery on the other end. So in order to address these questions about how natural history collections can help us understand something about the current pandemic or future pandemics, we need to keep in mind this concept of the extended specimen, and we need to understand that we need a robust cyber infrastructure that would allow us to connect all of these pieces of information. So let's think about what sorts of information we might need if we were actually going to use the power of natural history collections to address questions of the host, the origin, the transmission properties, and so on for SARS-CoV-2 or for any other virus or pathogen that we're interested in either mitigating or preventing. So at the center of this concept is our putative host. And here we will consider this a bat host. There, of course, have been other hypotheses about what the natural host might have been, but let's consider bats. And so we have, within our natural history museums, we have bat specimens. There are millions of bat specimens around the world in natural history collections. Now, using the information from the labels, we can find out information about the distributions of these different bat specimens, the different species, and so on. Now, in order to be able to link all of this information together, we actually need to make sure that we understand the taxonomy, or that we're using the same name for the same species in different collections and in different parts of the world. So the genus of bats, the horseshoe bats that are hosts to a large number of coronaviruses, including coronaviruses similar to SARS-CoV-2. This is the genus Rhinolophus. But there have been a number of taxonomic changes by specialists over the years. So have those names actually translated back into the specimens in the collections. There's also vast literature found in books, in particular about natural history of bats and other possible putative hosts. So that literature is certainly important. It's also important to have DNA samples and other sorts of tissue samples from the bat that can be preserved. And hopefully, if these specimens are processed, right when the bat is collected, we can also preserve the sequences of any pathogens. So hopefully there would be pathogens or potential pathogens, there would be parasites, other sorts of organisms that are actually found associated with the specimen. And then there would be DNA sequences, which may be found in various international databases. Again, information that would be accessible. And then there would be some analyses, data sets, and potentially new publications that could be accessed. So we think we actually understand what we need. We need host specimens, we need information about distributions, we need to make sure that our taxonomy is correct, we need access to the literature, we need samples of tissues so that we can look for pathogens, we need DNA sequences, and we need access to detailed data sets that have already been published. So through this task force, we investigated how available all of these pieces of information actually are. And what we located is that there are gaps. There are gaps at every one of these points. And so despite the fact that we've been working hard as a global community to digitize information and make information available for decades now, there are holes in our data at every turn. So this led the task force to develop some specific activities, some of which are ongoing. And what I'd like to focus on particularly here is just item number 3, which is develop a biodiversity related knowledge hub on COVID-19. And this knowledge hub would include information on all potential hosts, just as I described in those previous slides, as well as any information that could be brought to bear based on studies of the virus itself. So this biodiversity related knowledge hub for COVID-19 would allow us to connect information on viruses with information on putative hosts. The museum specimens would serve as a central focal part of this knowledge hub development. And it would also include things like the taxonomic treatments associated with them. And it would also include access to the published literature. Now, of course, most published literature is not available digitally, and so it can't be accessed as part of a global analysis or development of this knowledge hub. So fortunately, there are organizations such as Plazi in Europe that are focused on actually liberating information from the published literature, and in this case, revealing information on mammal host virus associations.
מה זה אוסטס? מה זה אוסטס? אוסטס.
So,
Okay.
מארחים.
So how can these natural history collections contribute? What have we learned? Well, as I mentioned, the data are mostly unavailable. The host information is not digitized, it's not aggregated. The taxonomy is not up to date. The data quality, for example, for locations and distributions is generally poor. The species interactions data are limited. And there's a big disconnect between the genetic data available for viruses and pathogens relative to their hosts. And this is because the sequences for the pathogens tend to lack museum voucher specimens, or the identifier of the voucher specimen is not present. So what this means is that all of these issues need attention. They need attention not just for the COVID-19 situation, but for any future pandemics. And all of these issues, fortunately, are receiving attention with greater emphasis on cyber infrastructure. So we published a paper last year focusing on how we can integrate biodiversity infrastructure into pathogen discovery and mitigation. And we identified 5 key elements for enhancing future resilience to such disease outbreaks. And these have to do with developing guidelines for keeping samples of both pathogens and hosts and for having them connected informatically. Developing metadata to, again, connect the physical specimens with the samples that are used for DNA sequencing, particularly of potential pathogens. We need an expanded global investment in infrastructure, both physical infrastructure and cyber infrastructure, to support archiving of these biological materials. We need new channels of dialogue and communication among specialists who, to this point, generally don't interact with each other, such as museum scientists and disease ecologists and public health scientists. And finally, the financial support and strong leadership from federal agencies, international partners, and private foundations is needed to, are needed to develop a proactive and multidisciplinary approaches. Now, we also published an article in The Conversation, and this is a public sort of outreach article, and this was picked up by a number of other news outlets around the world. And we're very happy to see that because we felt that this is an important message that needs to get out. But COVID-19 is just one societal problem, right? We have so many others that are related to biodiversity. So for example, the IPBES, the Intergovernmental Platform on Biodiversity and Ecosystem Services, has identified that these drivers of change have accelerated during the past 50 years. We've seen huge changes in land and sea use, exploitation of organisms, climate change, pollution, and invasive species. And all of these are modifying our ecosystem services and having very substantial impacts. So again, we need to use collections to address these societal challenges. So one of the possible solutions is to develop this robust interconnected infrastructure that links the data across museums and repositories. And this sort of infrastructure is currently being developed, but it's got a ways to go. We also need some innovations and implementation in order to connect the extended specimen with all of these additional pieces of information, like the traits, the phenology, the gene sequences, and so on. And we need tools and we need databases. So for this extended specimen concept to work, and this is certainly a foundation that will be important for applying natural history collections to pandemics in the future, we need to have all of these pieces of information linked together. And then they need to be linked to other sources of data so that we can extend the extended specimen. And this can be done through the development of what have been referred to as persistent identifiers, or computational ways that these different pieces of data can be linked to each other. And then through the use of artificial intelligence or machine learning to develop, we can develop new trait data sets that can also be used. And I showed this here because this was a recent special edition of applications in plant sciences. But the same sort of thing could be applied to bats and their habitats so that we can have more information that could be useful for the pandemic situation. And again, all of these data need to be open. We need development of new tools and cyber infrastructure. And through that combination, whoops, I think I missed something. Through that combination, we hopefully will be able to use our collections, not just to understand and mitigate, but perhaps even prevent future pandemics. Now, there's one final example that I'd like to present, which will only take me about 1 minute, I think, with regard to unanticipated uses of collections. And this has to do with the field called biomimetics, or bio-inspired design and engineering. It's amazing how engineers and inventors are using natural history collections, the physical collections in some cases, or the images in others, to develop new materials and robotics. So drag reduction of various materials is being studied by using shark skin. Adhesion of different materials to each other is being studied by looking at the toe pads of geckos. And optical biomimetics, such as the development of reflectors, is using iridescence that comes from a number of different sorts of organisms and structures, such as butterfly and moth wings, and also the iridescence of the marbleberry. So there are applications of these collections to biomimetics, and this is very important for a developing bioeconomy. So my key messages today are that natural history collections and associated data are important resources for the study of COVID-19, and that collections and biodiversity science can help predict and possibly prevent future pandemics. There are important lessons learned from our recent experience that can be extended to these additional issues. And one thing to keep in mind is that the reason we have been seeing an increase in the number of zoonotic or animal-borne pathogens and disease outbreaks over the past few decades is that we are continuing to impinge on natural ecosystems. And the more that we disrupt those natural ecosystems, the more likely we are that human populations will come into contact with various sorts of microorganisms that, while never pathogenic to humans before because they didn't come into contact with us, have the potential to be so in the future. So our natural history collections have important roles to play in addressing many other 21st century societal problems. But in order to do that, we actually need better integration of data and better development of cyber infrastructure to tie the data together. And this will involve the development of new tools and new methods through innovation and implementation. So finally, I'd like to acknowledge all of the folks who contributed to the many stories that I shared today, especially the National Academies Committee and the CTEF Disco Task Force, as well as the collaborators of our labs projects on the Florida climate change, phenology, and then my colleagues at IDIG Bio. So with that, I'd like to thank you all very much for your attention, and I'd be very happy to take any questions. Thank you.
Thank you very much. Fascinating. And well, the floor is yours and you can ask a question. Maybe I will begin with one question and someone will think about the other. So from what you have said, in the past, museum staff don't usually, in the US, I mean, don't usually cooperate with scientific or researchers regarding their collecting planning or what they are going to collect.
It's been very, I guess, idiosyncratic. So different collections. So each museum might have its own plan, but the museums hadn't necessarily coordinated among each other. And then the same thing would be amplified when you go from one country to another country. And this is where some of these global organizations like GBIF have been very beneficial in bringing together museum collections, natural history collections on a global scale, so that we can all talk to each other and understand what our priorities are, what our developments are, how we could go about improving the picture for the future. And so as a result, I mean, what's happened in the last 20 years has been pretty spectacular. And, you know, it's just kind of too bad that it didn't all start a bit earlier, but I think we are making some really great progress in developing a truly global community with shared goals and some shared resources. So I think that's very valuable. And I think an example of what can be done is this task force that had representatives from, you know, mostly Europe because it was founded by European organizations, but also North America. There were people from South America, people from Africa, and people from parts of Asia that all served on that task force. And that's really I think that's a novel approach when it comes to natural history collections, and it shows the potential for collaboration in the future.
Mm-hmm. Well, from the museum side, it's quite an, I think, a universal problem. The importance of collecting planning and to decide what is important and what is not, in general, in all subjects, art and history and whatever.
Yeah, I imagine that, and I was actually curious about this because it's not often that I get to interact with people from a number of other types of museums. But it seems like a number of the challenges that natural history collections face are also challenges that are probably common to other sorts of museums. For example, you know, underfunding for the work that needs to be done, not enough funding to cover conservation needs, you know, difficulties with upgrade and upkeep of facilities. You know, in the National Academy's collection that we focused on in that committee, we focused on natural history collections, but we found a very common pattern across all of those collections, whether big, small, focused on one little group, private, public, you know, and I guess I expect that those are challenges that extend across museum types.
Yeah, it is.
May I ask, what is the procedure if a collection, not necessarily a natural history museum, but a collection that holds botanical, not even a museum, a scientist that holds a botanical collection, historical collection, what will be the steps to join or to share the knowledge, the resources, the data? What would be the best way to do it?
Yeah, so probably, so it certainly depends on where one's located in the world, because there may be some national organizations to which, you know, with which you could share your data. But the other option would probably be to share the data directly with GBIF, the Global Biodiversity Information Facility. And that organization has a mechanism. So I'll back up a little bit, but there's a field of that has developed standards. So there are special ways, like to develop your spreadsheet that has, you know, like what you call your name, what you call your date, what you call your collector, and all of that. And if everybody puts their information into those fields using those standards, then all of that information can get compiled into one set without a person actually having to do it. And so the use of these standards, and they're called Darwin Core, has led to a mechanism that GBIF developed for basically sucking up data from other people's collections. So if you have a collection and you would like to contribute your data to something like GBIF, probably the easiest thing to do is to contact GBIF. So and their URL is gbif.org. So you can find them very easily.
You repeat, please?
Yeah, it's here, I can just put it in the chat, but it's
okay,
just,
great.
Oops, I can't spell. And there, I'm sure there's a place where it says, I want to contribute data, how do I do it? I'm not exactly sure where on the website you get to it, but it's something that's like that obvious. And you would contact them, and there would be someone who could actually tell you how to develop the spreadsheet in a way that you could share your data. And then so there's a method for doing it all in an automated way, but for beginners, there are also people who help manage that. So we have the same thing with iDig Bio, where we have a person who actually walks people through all of the steps if they need help, or if it's an organization that's done this a bunch, then they know how to set it up and they can contribute automatically. But you can contact GBIF and ask for assistance in contributing data, and somebody would help you.
Thank you.
Yeah,
that's very helpful.
Is there anyone else who has natural history specimens, whether plants or animals or fungi or anything?
I'm from, I'm the chair of ICOMNATIST, and I used to be a director of a natural history museum, and we had a conference about the issue of natural history collections contributing to disease spreading around the world. Not only, it was way before COVID-19. It was, you know, there were a lot of discussions about the necessity of keeping so much items in natural history collections, and we wanted to show that the importance of the natural history collections, not only as being exhibited to the people, but in research, all kinds of research areas. So we used it, and of course I agree with you that concerning the climate change today and the change of biodiversity, the museum collections are very, very important to understanding the process and how it's going to be looked like, and what we can do to try to prevent some of the changes. So I agree that it's very, very important.
Thank you for sharing that. Yeah, I think that some of my colleagues, for example, Joe Cook at the University of New Mexico manages a vertebrate collection, and their collection was instrumental in identifying the spread of a disease-carrying virus, the hantavirus. And they were able to, through their studies of museum specimens, particularly for small rodents, they were able to track the patterns of virus transmission, and the collections were incredibly useful for that. It doesn't always work, depending on the type of virus. The virus may or may not be preserved in the natural history specimen, and that's why it's important also to take genetic resources and have a genetics repository as well. So at New Mexico now they have, I think they have close to 250,000 genetic samples from individuals. So many of the individuals they have in their collection as the physical specimens also have materials that could be used for genetic analysis.
Yes,
and going into the future, that would be ideal. Oh, sorry, Dorit.
The Hebrew University in Jerusalem, we have a very good lab for taking DNA samples for natural history collections, and the head of this laboratory, she's a member in SETAF.
Oh, okay.
She's doing a lot of research. Yes.
Okay. Oh, great. Thanks for sharing that.
I say a word in Hebrew because האוספים הלאומיים של ישראל במדעי הטבע מחולקים בשתי אוניברסיטאות, אוניברסיטת ירושלים ואוניברסיטת תל אביב, ובאוניברסיטת ירושלים פרופסור גילה קחילה מנהלת מעבדה שלוקחת דגימות DNA מאוספי טבע, ויש לזה חשיבות מאוד מאוד רבה, כמו שהמרצה שלנו הסבירה, בהבנה של התפזרות נשאים. קודם מישהי שאלה מה זה הוסט. הוסט זה נשא. הנשאים פוטנציאליים למחלות ויראליות. זה מאוד מאוד חשוב, כמו יש מחלות, כמו לדוגמה מאוד מאוד עתיקה, דבר למשל, כן? שהוא עברה דרך מחסמים. אז יש לזה מאוד מאוד חשיבות מאוד מאוד גדולה להבין ולחקור את הנושא. I was explaining what a host means because earlier someone asked about what is a host. So I explained something. It's the animal who carries on the virus from one, might be from from the animal to humans, or from one place to another. So that's what I explain in ebook.
Oh, thanks. Yeah, I guess I should have explained that earlier. So thank you for explaining that.
Yeah, I think it's more or less have been understood from your lecture. And if I will return to the collecting planning, in your museum, in the Florida Museum, do you have a collecting planning for deciding what to collect and not?
So, so different collections have different goals. So, and part of that is historical. So the herbarium that's part of the Florida Museum has been focused on the southeastern United States and the Caribbean, and that's where most of the collecting is focused. But other collections, such as our invertebrate zoology collection, has a large collection of marine organisms from the South Pacific and the Indo-Pacific. And some of these collections, it's sort of the history of who the scientists were who were working there. And, you know, so these collections have kind of developed until more recent times, sort of haphazardly. But what so our geographic regions are not all connected, but what we do have is a connected, we have separate databases for each collection, but they're all in the same system. So that if we pulled out if we found a plant species and we wanted to know what other species from the other collections occurred at the same location, we could find out that information through our connected databases. We also have a connected genetic resources repository, and that collection has samples from all of the separate collections, so that again, it would be possible for more recently collected specimens to investigate the genetic variation among different species from different groups of organisms in a particular geographic area. We've been talking about potentially having a new focus just on Florida because of the ways that climate change is expected to have such severe impacts on Florida. So when, you know, a small state being surrounded on three sides by water with sea level rise, such an important future development, and plus changes in temperature and precipitation, we think that it could be very valuable for us to have a more focused collection plan just in our own region. But these are the types of, but of course, to do anything like that requires like new funding. It's like, and most institutions can barely, I don't know what your institutions are like, but the case here is that we can barely, you know, maintain, let alone develop new programs. So except for the digitization, because that was a big investment by the National Science Foundation at a national level. But otherwise, yeah, it's pretty difficult to develop any new sorts of programs.
Yeah, my specific museum is based on donations, so it's pretty different.
Problems with invasive species in Florida.
Invasive, yeah, so invasive species are also one of our major issues, and that certainly transcends the type of organisms. So we have many invasive plant species. There are a lot of, there are invasive lizards in South Florida in particular, some huge invasive snakes. There are invasive fishes that are moving into the marine waters that are destroying some of our marine habitats. And yeah, so invasive species are definitely a focus of research across the state and using our specimens to the extent that they can. So yeah, very good point.
That's a big issue in Israel because of our geographical constellation. We have invasive fishes from the Red Sea to the Mediterranean through the Suez Canal. And we have a lot of birds invasive. אנחנו מדברים על, I will say it in Hebrew, אנחנו מדברים על מינים פולשים לישראל. יש הרבה מאוד מינים פולשים, כמו מיינות למשל, שזה מין פולש. יש הרבה מאוד דגים ששחו מים סוף דרך תעלת סואץ וחדרו לים התיכון, וכתוצאה מכך הם גרמו להיעלמות של מינים טבעיים שהיו חיים פעם בים התיכון. וזה נושא מחקר מאוד מאוד חשוב במוזיאוני טבע. Sorry that I explained in Hebrew, but since our members not coming from natural history museums, some of the words that we use are not familiar with them. So,
yeah, thank you.
May I ask something, please?
Sure.
My name is Ruth. I'm from the world of library and archive. How about rights for herbaricums? You know, you get a collection from someone, you have to mention his name, the collector's name. Is it open for public? How do you manage that?
Sorry, so I got a little static right when you were speaking, but were you asking about the collectors when we get new specimens? Is that what?
No, I meant sometimes someone donate his collection to the museum. So usually it comes with conditions, you know, like name have to be mentioned or can be used in public, not be used only after 70 years or whatever. It's in the world of books, I know. Is it the same in herbariums?
Oh, thanks. Yeah, that's a really interesting question. Most collections, I think now that are being, or even over the last few decades that have been donated, I think when the donation is negotiated, the host in the institution has been able to negotiate to make sure that the information can be public. But certainly in the past, I mean, and here's a funny example of this. At the University of California at Berkeley, there are actually two separate herbaria that have to be managed separately. One is the University Herbarium and one is the Jepson Herbarium. They both focus on the plants of California. They both have plants from all over the world. And yet the Jepson Herbarium was donated by Jepson with an endowment that said this herbarium has to be, you know, curated in perpetuity, separate from the university one. So here at one of our leading institutions in the US in terms of herbaria, at least, and museums overall, but the herbaria are actually curated separately because of that issue with the donation and the endowment. So, and that of course makes things very difficult on the people who work there. It's possible to have all of the information public, but they still have to keep everything separate. So I think that's a good example to us of why we want to make sure that we negotiate our way out of those sorts of issues when we're preparing to accept a donation. Yeah, it's a very, very important point. Thank you.
Okay. So I would like to thank you again. That was interesting, and hope we could cooperate in the future. So thank you for everybody for participation, and
thank you. Great. Yeah, thank you again for the invitation, and thanks to all of you for your attention, and thank you for your questions. And I hope all of your museums open up again and are successful post-pandemic. Although I suppose, given what I read about Israel and your vaccinations, maybe things are quite open there at the moment,
yes,
given the fact that, yeah.
Most of all the museums are open now.
Great.
yeah.
Thank you very much. It was great. Thank you.
Yeah, thank you. All right, thanks again. Bye.
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 Was very helpful.